Okay, so we are officially being recorded for this live Zoom meeting / lecture. It's the first one on the muscular system. Quick reminder, you have a quiz today from noon until 11:59 p.m. It's short and sweet. It's not difficult. It's just over the business, the rest of the skeletal system and joints. But we do need to get into this muscular system business because it gets more complicated. Let me admit, Allison. It does get more complicated as we finish this course, this A&P1 course. So like I said on the first day of class, or at least the first meeting, I did say this, I'm going to repeat it, that things get more interesting as you go through this course of A&P, both A&P1 and A&P2. In my opinion, things get a lot more interesting, but they also get more complicated. So right up front, please take note. Expect these last two organ systems to be a little more difficult, a little more complicated, but hopefully more interesting as well. They'll be more relevant. So without further ado, I'm going to get right into the muscular system. And it's going to seem like I'm going a little bit more quickly than usual. That's because there's more ground to cover than usual. So I'm just going to jot down notes and speak. I'm not going to have time to write everything because I'm going to draw complicated pictures today, which will take time. So going to my first whiteboard, this is the start of the muscular system. So just remember there's 3 types of muscle tissue. And I will ask you or expect you to know a few characteristics about those 3 types. That's supposed to be a U. Just a quick review. There's skeletal muscle, cardiac muscle, and smooth muscle. And as another quick review, the characteristics, the brief characteristics that we learned when we were doing the tissues in a previous section. Skeletal muscle. Has striations. It's voluntarily controlled. The cells, each individual cells, are multinucleate, have more than one nucleus. And the skeletal muscle, remember the location, it's the stuff that's attached to bone. Skeletal muscle is the stuff that you and I think of when we hear the word muscle. A couple perhaps new characteristics. The cells are called fibers. Skeletal muscle cells, we call them muscle fibers. So if you hear me say the word fiber, muscle fiber, or you read the term muscle fiber, that means an individual skeletal muscle cell. Also, in skeletal muscle, last thing I'm going to say right now about them is each cell, this is important, each cell must be neurally stimulated in order to contract. All right, so each individual cell must have its own nerve branch ending in order to be stimulated and do its contraction. All right, second type of muscle is cardiac muscle. This is repeat, only found in the heart. It's also striated, it's involuntarily controlled, and it can have 1 to 2 nuclei per cell. Here's a worth noting, an asterisk. Cardiac muscle does not need neural stimulation in order to contract. So that's sort of a specialty with cardiac muscle. It can the fibers, the muscle individual cells can contract on their own without neural stimulation. You'll learn all about that when you study the cardiovascular system. Third type of muscle is smooth muscle. Here's some repeats. The location is it's found in our visceral organs and hollow organ walls, also lining vessels, arteries and veins. Smooth muscle has no striations, no stripes when we look at it under a microscope. That's why it's called smooth muscle, by the way. Smooth muscle is also involuntary, like cardiac muscle. We don't have conscious control over it. Smooth muscle only has 1 nucleus per cell. This is all in your book, by the way. Smooth muscle is also sometimes called visceral muscle. That's just because it's found in the walls of visceral organs, mostly our digestive organs, also vessel walled. Now, smooth muscle needs neural stimulation to contract, but each individual cell does not need its own individual nerve stimulations. Smooth muscle is able to transmit electrical stimulation from cell to cell. Skeletal muscle can't do that. All right, so that's 3 types of muscle tissue. That was all in your textbook. I just wanted to reiterate those, and I'll just ask you a couple questions about that in the next quiz. Next, I'm just going to list characteristics, or I'm going to state characteristics of muscle tissue. So this is something that you need to know. This is, by the way, this is muscle tissue in general, not any particular muscle tissue type, but it's muscle tissue in general. First one, I'll write these out. I'm not going to write out the explanations. First one is called excitability. What that means is this tissue is able to receive and respond to a stimulus. So it's usually a chemical stimulus from the nervous system, but excitability means it's responsive to some sort of stimulation. Okay, the next characteristic on the list, you know this already, is contractility. That means the cells, the tissues, the cells in the tissue are able to shorten or contract. So when they're stimulated, they're able to shorten and contract, and as a result, usually produce some sort of tension or energy. So we've got excitability, contractility. Next word is extensibility. A lot of these words sound the same, but they're different. So it sounds like excitability, but extensibility means these cells or tissue are able to extend or stretch. So think of extending, stretching. Next characteristic is elasticity. That's sort of after, these two go hand in hand, by the way. After a cell can stretch, it's also able to recoil to its original position. So after it contracts or stretches or extends, cells are able to recoil or snap back to their original, that's what elastic means, recoil, to snap back to their original size and shape. This one, contractility, is the true unique one from other cells. All right, now I'm just going to list major functions. This is of the muscular system, not just of muscle tissue, but of the muscular system, this organ system. And this system produces movement because in general, when a muscle cell shortens or the muscle tissue shortens, that produces some sort of movement. When you and I think of skeletal muscle, it's attached to our bones. It pulls on those bones and our arms move, our legs move, but also inside of us, vessel walls move, organ walls move, the heart moves due to contraction of the tissue. It also allows us to maintain our posture. You're probably sitting down right now listening to this lecture. Well, there are muscles in your hip region all the way up your back that are contracting, allowing you to sit up straight. The muscular system in general stabilizes joints. That's supposed to be a T. Muscle tissue can also regulate temperature. I'm going to put generate heat because that's what your textbook says. It helps us regulate temperature, body temperature. You know, when you exercise, that generates heat, warms up your body. Also, if you walk outside on a cold day, you start to shiver. Those are tiny muscular contractions that also generate heat. A few added functions that are, I don't know if they're in your textbook or not, but muscles can provide protection. Your and my abdomen, the organs in our abdominal cavity, don't have a lot of bones around them for protection. It's the muscles in our abdominal region that actually protect those organs. So protection is one. Regulation, I'm just going to put regulate valves. Inside of us, for example, in our digestive system, we have sphincters and valves that regulate the flow of food from one area to another. And those valves and sphincters are typically made up of skeletal muscle and smooth muscle that can open and close. And finally, muscle cells can store nutrients. I'll talk about that a little bit later. Usually in the form of glycogen, which is a stored form of sugar. All right, let's move on. Now I'm going to talk about, pretty much from this point forward, for the next couple days, when I say the word muscle, we're going to only be talking about skeletal muscle. So, and most of your quiz, the next quiz is going to be about skeletal muscle, a little bit about smooth muscle at the end of this unit. Cardiac muscle, I am not going to talk about after this sentence. The reason is because when you take AMP2, you'll study the cardiovascular system and you'll study the heart and cardiac muscle in much more detail. The majority of this section is about skeletal muscle, and we will talk about smooth muscle at the end when we have time. So if I just say muscle, now you know I'm talking about skeletal muscle. That's the stuff that's attached to bones that we voluntarily can control. Every muscle fiber cell, I'll say fiber cell for a while, but after that, if I just say fiber, I mean an individual cell. So every muscle fiber cell is supplied with its own nerve ending. I already stated that, for stimulation. That is unique with skeletal muscle. So I'm just going to write has own nerve ending, each individual fiber. And it has to in order to contract. Skeletal muscle fiber tissue and fibers also have a very rich blood supply. You know that these muscles, these skeletal muscles, are what make us active. You exercise, you walk, you run, you climb stairs, whatever it is that you do, that requires a lot of energy. So that requires a lot of nutrition, high oxygen supply, and those muscle cells also produce a lot of waste that need to be transported away from the muscle cells. So very rich blood supply. Now I'm going to write macro anatomy. So we're going to look at the anatomy of a muscle, how it's made up, without any microscopic assistance. When you just look at a muscle, this is what you're actually looking at. I'm going to switch my pen to red, and I'm going to draw. Muscle fiber cells are long and cylindrical. That's why they're called fibers. And they look like this. So this is an individual cell. They're sort of tubular looking. They have connections at the end, but I'm going to cut off the connection. And there's an individual fiber. Each individual fiber or cell is wrapped in a connective tissue covering, and that has a name. A very thin, delicate connective tissue covers each individual cell. And that thin, delicate connective tissue covering on each cell is called endomysium. Okay? Now, how a muscle is arranged goes like this. Each individual cell that is wrapped in this connective tissue covering, they get bundled together, these individual cells. Think of, if you're at Home Depot and there's a stack or a pile of PVC pipes or tubes, small in diameter, that are sort of bundled or stacked together on a shelf. That's what these look like. Now we bundle these. Now each individual cell is wrapped in endomysium. I'm not going to draw that because it would just take too long. We can then bundle a group of individual cells together and wrap those in a different, thicker connective tissue covering, forming a bundle. This thicker connective tissue is called perimysium, and it wraps several individual fibers together to form a bundle, which is called a fascicle. It's spelled F-A-S-C-I-C-L-E. That's just, when you see that word, just think bundle. That's all it means. It's a bundle of muscle fiber cells, a fascicle. And what makes up a fascicle is it's a bunch of fibers wrapped in a thicker connective tissue called perimysium. Now, if I were to take, I'll draw quickly so it'll be a little messy. If I were to take now several fascicles, so I drew one fascicle, now I'm going to make another fascicle here that's wrapped in another perimysium. So here's another fascicle, and I'll make another one up here. These are individual muscle fibers that are wrapped in endomysium. Now I'm going to wrap them in perimysium. I've made 3 fascicles. Here's another one. If I wrap a whole bunch of fascicles in a super thick connective tissue covering, which is what I'm going to do now, this is an entire muscle that I've created. This super thick connective tissue is called epimysium. It's coarse connective tissue that wraps several fascicles together to form an entire muscle. And that's how our muscles are arranged, our skeletal muscles in the human body. We've got individual fibers wrapped in endomysium. We bundle those together with perimysium to form a fascicle. Then we wrap several fascicles together in epimysium to form the entire muscle. All right, now we're going to move on. Now I'm going to go the opposite direction and talk about microscopic anatomy of muscle cells. So what I'm going to do in the next board that I draw is I'm going to take one of these individual cells, and we're going to look at what's inside of that, okay? So let me layer on here. Now I'm just going to draw one big cell. This is an individual muscle fiber. I'm leaving space on the right-hand side for a reason. You know that this is wrapped in endomysium. I'm going to leave that off for now. We're going in with a microscope and we're going to look inside of this fiber. So this is arranged almost identically to the picture I drew back here. This picture that we just drew is just a whole bunch of tubes wrapped together to form bigger tubes, wrapped together to form larger tubes, yada, yada, yada. And what we're on the inside of this individual cell, it's also just smaller tubular structures within each other. So inside of this individual cell are these long cylindrical organelles called myofibrils. I'll write that out. And the muscle fiber cell is just packed with these rod-like structures called myofibrils. M-Y-O, that's not an X. Let me do a better job of that. A myofibril, that's an R-I-L. M-Y-O-F-I-B-R-I-L. This is an organelle, a specialized organelle inside of each muscle fiber cell. It's a long rod-like structure that contains smaller rod-like structures called myofilaments. Before we go any further, this prefix, myo, just means muscle, okay? That's its definition. Also, this prefix, sarco, we use that when we talk about muscle as well. So whenever you see this, myo, just think we're talking about muscle. And so the myofibril literally translates to a fiber-like structure within a muscle. So this is a specialized organelle that contains myofilaments, which are these thinner rod-like structures. So inside of each myofibril are these thread-like structures called myofilaments. Okay. I'm going to go through in this microscopic anatomy now. Now you have the gist, at least in your mind, of how an individual muscle fiber is arranged if we look inside of it with a microscope. I'm going to write some terms and I'll define them verbally for you, and we're going to construct how we're going to examine what goes on inside of these myofibrils. And at least our goal today is to determine sort of how a muscle cell contracts, how it carries out the act of contraction, okay? So let me later on a new page. If you need me to go back to one, just ask. I'm going to do some terminology here. We need to build a dictionary so that when that's S-A-R-C-O-L-E-M-M, that's supposed to be two M's, A, sarcolemma. This is just the plasma membrane of a muscle fiber. So it's just the outside covering. I'm just going to write plasma membrane of a muscle fiber cell. So that endomysium that wraps an individual cell would be on the outside of the sarcolemma. This is actually part of the cell. It's the plasma membrane. Next word is sarcoplasm. This is just the cytoplasm of a muscle fiber. Sarco means muscle, plasm, just think of cytoplasm of the muscle cell. It contains things like glycosomes. Let me write cytoplasm. It contains glycosomes, which are granules of stored glycogen, stored energy, stored sugar. It also contains myoglobin. This is a muscle form, myo, of hemoglobin that binds oxygen. We have hemoglobin in our blood that binds oxygen. Myoglobin in muscle cells also binds and stores oxygen. And it, just like hemoglobin, when oxygen is bound to it, it turns a bright red. All right, we already figured out what a myofibril is. It's a rod-like organelle inside. It's a specialized organelle that's rod-shaped. The organelle that contains thread-like structures called myofilaments. These take up most of the volume of the cell, by the way. That's why I drew my muscle cell containing tons of about 60 to 70%, maybe even up to 80% sometimes, depending on the muscle we're talking about, is just the volume on the inside of the cell is just these long rod-like structures. So I'm going to get more specific about myofilaments now. They are within the myofibrils. These are smaller, I said thread-like before, but if we magnify them enough, they're also rod-like. And they're peptides is what they are. That just means a long chain of amino acids. But they're smaller rod-like structures inside of the myofibrils. And there's 3 different types of myofilaments. There is myosin, which we call the thick filament. There is actin, A-C-T-I-N, which we call the thin filament. And there's a third one that doesn't get much press or much advertising. It's called the I'm going to write, its name is titin, T-I-T-I-N, and it's known as the elastic filament. Remember, elastic allows for recoil, whenever you see that word. That's actually how a cell is able to snap back to its original size or its original length. So we're building a dictionary here, okay? So I'm going to add on to this word. Sarcomere refers to, you'll hear me say this later, it refers to one contractile unit of myofilaments. I'm going to draw some pictures in a minute to show you how the inside of a muscle cell is arranged. But I'm going to use this language, and I want you to be able to refer to it. So we mentioned myosin and actin. I'm going to describe them in more detail, slightly more detail now. Myosin, we know that that's the thick filament. It's a long polypeptide chain. It has a globular head with a long tail. And it's just a chain of amino acids. The heads of these form what are called cross bridges with actin molecules, the thin filament. The heads also have the ability to bind ATP. That's where a muscle gets its energy from this molecule, which is why we mentioned it the first week. A muscle cell gets its energy in order to contract from the ATP molecule. And it cleaves that terminal phosphate off the end of ATP, and that's what gives the muscle its energy to contract. Actin, the thin filament that we've defined earlier, it's sort of a smaller globular chain, peptide chain. I'm just going to put amino acids. It's a smaller globular chain of amino acids. It's described, it looks like beads, a necklace, a beaded necklace that's just laid out straight, not in a circular fashion. Actin molecules have active sites on them, so that myosin have active sites for myosin to bind. New term. After I define, it's not going to be that much longer, after I write these words out that are important for you to know and understand, and the full definitions of these are in your book. Tropomyosin is also a polypeptide strand. That just means a chain of amino acids. Peptide is just a long chain of amino acids. So it's a polypeptide chain or strand that blocks the active sites on actin. Blocks the active sites on actin. That's so that myosin cannot bind to actin. This is the situation when our muscles are relaxed in a non-contracted state. One more word and then I'm going to switch panels here. This looks like tropomyosin. It's pronounced troponin, T-R-O-P-O-N-I-N. This is a small, very small, it's only 3 polypeptides, I believe. Small 3 polypeptide that is attached to tropomyosin, the longer polypeptide. And this troponin binds with, this will be important later when I draw a picture, binds with calcium ions. So I'm putting Ca++. That's how I will write calcium ions from this point forward. So a few more definitions now, then I'm going to draw a somewhat elaborate diagram to help you understand how all of these things work together. Okay, so let's layer on one more board. All of this, by the way, is in your textbook. If I'm going too fast or you need extra explanations, that's supposed to be a lowercase a. Sarcoplasmic reticulum. Sounds a lot like endoplasmic reticulum, that one of the organelles we learned in a cell, and it's somewhat similar. What it does in a muscle cell is this is basically a set of tubules. It's a massive set of tubules that has big pouches associated with it, but it's a set of tubules around those rod-like organelles called myofibrils. So we're still inside of an individual cell. It's a set of tubules around the myofibrils. It has these pouch-like structures called terminal cisternae. So I'll describe those. Pouch-like structures that store calcium ions. Okay. This sarcoplasmic reticulum is also closely related, physically, I mean touching, with T-tubules, and I'll define what those are. So we know that the sarcoplasmic reticulum, here's its main function. It stores intracellular calcium. I'll draw a nice diagram of how all of these things relate to each other in a moment. I have 2 more things to define here. T-tubules, that's short for transverse tubules. That's where it gets its name. These structures are closely related. They're elongated tubes that conduct, I'll write this out, they conduct electrical impulses It's a C. From the sarcolemma, the plasma membrane, to the inside of the muscle fiber. Last term, I promise, and then I'm going to put all of these terms together for you in a picture. I'm going to use this terminology a little bit. Triad. This is not a specific structure. This is a name that we use to describe two terminal cisterna of the sarcoplasmic reticulum. So I'm just going to write of the SR plus one T-tubule. Okay. Now I have two pictures to draw and then we'll be done for the day. The first picture I'm going to draw, and then I'll draw even a more elaborate one after the first one. Inside of a myofibril, so I'm going to draw a rod-like structure. This is deep within one cell, okay? So this rod-like structure is one myofibril. That's an organelle, a specialized organelle within each muscle cell, and there's hundreds to thousands of myofibrils in each cell, believe it or not. So one myofibril, we're going to look at first the arrangement of the myofilaments within that myofibril. So this is what I'm going to draw here is the pattern of myofilaments. There's all sorts of pictures in your book with this. I'm going to try to simplify it for you so that when you go to look at those pictures, you'll have an idea of what you're actually looking at. All right, so I'm going to draw first is a straight red line this way. It's kind of hard to do on a vertical glass surface to draw a perfectly straight line. I need to draw sort of a stem in this direction. Now I'm going to draw the thick filaments. They emanate out from this thing and they have a globular head on them. They're not arranged quite this neatly. So these are myosin molecules, those myofilaments. The thick myofilaments are what I'm drawing right now. They have a globular head and a long tail. They kind of remind me of the shape of a golf club. A golf club has a shaft and then a head at the end of it for hitting the ball. Not that I'm a big golfer, but I know what a golf club looks like. That's what these kind of remind me of when I draw them every time. And technically, they're not quite arranged. They're not singularly arranged. Inside of a muscle cell, we have a myosin molecule, and then right next to it is another myosin molecule. They bundle together like this. I'm not going to draw that because it'll just mess up my picture. So I'm just going to draw one thick filament. I'll label it for you. So this is myosin. Remember, we're inside of a myofibril. Now I'm going to draw the actin molecules, and I think your book typically uses blue for that. I'm used to drawing them in yellow, but I'm going to use blue because that's what your book does. The thin filament kind of looks like this. They're close to, but not touching. Sorry, I shouldn't have touched that. I didn't try to do that. They're close to, but not quite touching the thick filaments. The thin filaments are arranged, they're held together by strands like this. I'm going to draw, it repeats over here, the exact same mirror image. And now I'll label these thin strands. That's actually an actin molecule, the thin filament. And this pattern, what I've drawn here, so from here to here, this thing, this blue thing right here is called the Z disc. And it's just what holds the actin molecules together. From one Z disc or Z band to another is called one sarcomere. And that is one unit, one contractile unit within a muscle cell. So from here to here is a sarcomere. That's one contractile unit, and it repeats then over and over again, hundreds and thousands of times. There's new actin molecules coming off of the Z disc or the Z band in this direction. And there's just more thick filaments associated with those like this, and they get held on a new M line, this stem. I'm not going to draw them quite as neat because I'm in a hurry. And there would be another repeating pattern over here, and it just seems to go on forever. If you look with a microscope inside of a muscle cell. But keep in mind that this is one contractile unit. And here's how, this is the short story of how a muscle contracts. This is the goal, the end of the story. What's going to happen when a muscle contracts is this myosin head is going to reach up and attach to its corresponding actin molecule, the closest molecule. So it reaches up and grabs the actin molecule. And then because the myosin head has energy from ATP, that myosin head is going to pull that actin molecule toward this red center line, and that's called the M line. And these myosin molecules are going to pull these corresponding actins toward the center line in the opposite direction. And that happens in every single contractile unit all the way along a muscle fiber cell, hundreds to thousands of times. So these myosins, when a muscle contracts, reach up and grab these actins and pull them this way. And the ones over here on the left reach up and grab their actin molecules and pull them. I'll make this one grabbing down. They pull actin and that toward their M line. This just repeats. There'd be more myosins this way, and they would pull their actin molecules toward their M line like this. And you can see now when this happens, the entire muscle fiber cell is going to get shorter because this happens over and over again, hundreds to thousands of times within one cell. And so the overall cell is going to go from a long to a very short, usually thicker, a short state like this. I can't draw down below this line on my screen. So that's really what's happening. If you have that in the back of your mind, that these thick filaments, these myosin filaments, they reach up and grab the thin filament actin and they pull it toward the M line. It's the actin molecules that slide and move. The myosin molecules stay put, they stay stationary, but the actin molecules actually do the sliding in this direction and this direction, and that's why a muscle shortens when it contracts. So that's the first drawing. The last drawing I'm going to show you is my last page, I promise. I need to clean some things up. You know what? Tomorrow, when I start this talk, our next talk, I will redraw this page and label a few of these zones in here. I did label the M line, but I'll remind you, and I told you that this is called the Z disc here, but there are other banding patterns within this. Where we have no, for example, where we have no, in this area here where there's no thick filaments, we call that the I band. And when there's overlapping myosin and actin filaments, we call that the A band. I'll clean this up tomorrow and draw all of those banding patterns for you. The last picture I'm going to draw today, we're going to lessen our microscope magnification. The last one that I just drew, this picture, is super high magnification within 1 myofibril. The next one that I've just started to draw right here is going to, this is the entire muscle cell. I'm going to show you how, so I'm going to write 1 fiber. That vocabulary dictionary that we just created, I'm going to sort of show you the arrangement of how these things work. There's a nerve cell ending that stimulates this fiber cell. I need to redraw this, the top of this cell. There's an indentation like this where the nerve, so I'm going to write neuron, meets the actual muscle fiber. I'll explain this to you more than once, so don't feel like you have to grasp this all today. And I'm going to do it in more detail tomorrow. When a nerve cell fires an electrical impulse, it travels down to the end of this nerve cell. The nerve cell releases a chemical. This is the short, easy version because I'm going to layer on more complicated steps as we talk about this further. A chemical is released into this space. It has a name that I'm not even going to say yet. That chemical binds to receptors on this area of the muscle fiber cell. So what a nerve cell does when it, quote, fires an electrical impulse, it releases chemicals. That chemical binds to the surface of the muscle fiber cell, causing the muscle fiber cell to generate its own electrical impulse along the surface of the cell. Now we're going to use some of the language that we conjured up in our vocabulary list. That electrical impulse is guided inward down these structures called T-tubules. So that's what I'm drawing now. These blue things are called transverse tubules or T-tubules. The T-tubule's job, its function, is to guide an electrical impulse inward to the inside of the muscle cell like this. At the base of each T-tubule is a sac-like structure called a terminal cisternae that is storing calcium ions inside of it. And I'm drawing 2 terminal cisternae associated with each T-tubule. That creates a triad. So this is 1 terminal, sorry, that's supposed to be a C. This is 1 terminal cisternae. There's no E at the end if we just are talking about singular 1. Terminal cisterna is coupled with a T2. So we have 2 terminal cisternae. There's one and there's another one. Associated with each T-tubule, which is in blue. this thing, sorry, I thought I had yellow. The terminal cisternase function is to store calcium ions. When the electrical impulse comes from the nerve, causes chemical to be released, the chemical binds to the surface of the muscle cell, that generates another electrical impulse along the surface of the muscle cell. The T-tubules guide that electrical impulse inward. When the electrical impulse reaches the triad, a triad is just two terminal cisternae associated with one T-tubule. So when the electrical impulse gets down here, it causes the stored calcium to be released from the terminal cisternae. So we've got, I'm going to say this one more time and then I'll continue tomorrow. We've got a nerve impulse, electrical impulse, causes chemical to be released. The chemical binds to the surface of the muscle cell, muscle fiber cell. It binds to the sarcolemma, that's the plasma membrane. That generates an electrical impulse along the sarcolemma of the muscle cell, the plasma membrane. The T-tubules guide that electrical impulse inward. When the electrical impulse reaches the triad area, which is just two terminal cisternae associated with one T-tubule, calcium ions are released from the terminal cisternae, and the calcium ions flood the inside of the cell. I'm going to draw this picture tomorrow, but just so you know, since you guys attended this live Zoom meeting, I'm going to tell you, I'm going to go back to this picture. What calcium is going to eventually end up doing is it's going to allow myosin to bind to actin, and make all of these binding sites, so that then myosin can pull actin toward the M line. So the whole point of releasing calcium is to allow myosin to bind to the actin molecule. So with that in mind, that's our final picture of the day. The first thing I'm going to do tomorrow is redraw especially this picture, and I'm going to layer on some detail. But now that you know the basics of it, the gist of it, when you read your textbook and when you listen to me lecture tomorrow, you'll have a better idea of how a muscle cell contracts, okay? So I'm going to stop recording now.